Pre-Med Peers. I'm excited to work with you guys.
So just a general introduction about, we have MCAT 101 here, but we're transitioning to the name Pre-Med Peers. So sorry if that is confusing, but we're registered as a nonprofit in Virginia, and the primary goal of this organization is just to pass on what we learned about the MCAT because it's such a convoluted process. So we hope that we can be accessible to you via like Slack and email. So don't hesitate to ask us any questions that you have, even if it's like tangentially related, that's totally fine. So all our sessions are recorded. This one is currently being recorded. So if anyone has a problem with that, please do be aware that it is being recorded. And then that also helps you refer back to it later. And then don't try to worry about, I'm so sorry, I don't know what's happening. Don't try to worry about like getting all the notes down and having everything like processed in your brain at this moment. You can always refer back to it. And our email is now info@premedpeers.org. So if anyone has been in like past topic session, we had a different email. So please note this as well. And then if you are not on the newsletter or listserv, either put your email in the chat or so that we can add you to our listserv. And then before we get started with our actual lesson, does anyone else have any questions? Okay. So the setup of today's lesson is that we're going to review the homework questions that come from the end of the chapter in the Kaplan books. And then we have discrete warm-up question and a topic presentation, which will be like a very brief summary of what you learned in the books, as well as the discussion. And then at this part, you can also just ask questions about what you struggled with. And then we have a passage practice. So we have a passage as a PDF that has 6 questions on it today. So hopefully we'll have time for that. And then at the end is like another general Q&A and our conclusion. So please don't hold back any questions. And hopefully this can be interactive and technical problem-free. Right. So this is an overview of the chapters. I'm not going to read them out loud, but we just put it here so you guys can refer to like what the lecture is made out of if you're going to look back on it. Okay. And then I'm going to, Karen, do you want me to stop sharing and then take the screen back?
It's okay, because you can just leave it here, because this is the part where like I just ask if anybody has any questions. Okay. And if someone has questions on a specific question, I can just I can share my PDF of the book.
Okay, sounds good.
So does anybody have any questions on the homework? How many people did it?
Could we go over number 6 from chapter 6 questions?
Number 6 from chapter 6. Okay, so let me pull that up on the screen. Okay, so I'm in chapter 6 already. Can everybody see my PDF?
Yeah, there's loading right now.
Okay.
Now it's good.
Okay, so number 6 on chapter 6 is which of the following enzymes is not involved in DNA replication? And so we have A, primase, B, DNA ligase, C, RNA polymerase 1, and D, telomerase. So the answer to that question was C, so RNA polymerase 1. The reason why that's the correct answer is because it's actually involved in transcription, not replication. Does that make sense?
Yes, thank you.
Okay. Anybody have any other questions on any of the questions?
I just have one, but so I wasn't quite sure, but if it's anything like biochemistry, like homework that was supposed to be assigned, I'll be honest, I didn't do them because I never took biochemistry before. So I don't know if I should do the biochemistry questions without prior knowledge to biochemistry, or just wait until I take the class first, because I don't know how people do it. Because it's like if you wanted to study with the Kaplan book, or you want to do Khan Academy on biochemistry, that's why I didn't do the homework. I know we're supposed to do it, but if someone never took biochemistry before, I wouldn't really know how to start in the beginning.
Okay, that's totally fine. We completely understand. Let me admit this person first. So I guess it'd be best moving forward. I guess you can, we're going to be going over kind of like the basics of what everyone else should have gone over already. And it'll be about like 20 to 25 minutes of kind of the basics. And then from there, hopefully it'll be easier for you to either go through the book or Khan Academy. And then you can do the practice questions if you'd like.
All right, thank you so much.
I had a question on number 10 for chapter 7.
Number 10 for chapter 7?
Yes.
Can everybody see my screen? Just making sure.
Yes.
Okay. You said number 10, correct?
Yes, number 10.
Okay, so this is a tough one. If you guys got this wrong, it's completely understandable. This is not the easiest question. Okay, so a 4-year-old toddler with cystic fibrosis is seen by his physician for an upper respiratory infection. Prior genetic testing has shown that there has been a deletion of 3 bases in exon 10 of the CFTR gene that affects codons 507 and 508. So if you can see my mouse, this is where the XXX is where the base pairs are missing. So what effect will this mutation have on the amino acid sequence of the protein encoded by the CFTR gene? So this is actually a 2-part question. So they're asking you, number 1, which residue is going to be deleted, phenylalanine or leucine? And the second part of the question is, does this change the C-terminus sequence, in other words, the end of the sequence? So the answer to that question, give me 1 second. So the answer to the second question is A. So I'll go over the second part of the question first. So the reason why there's no change in the C-terminus sequence is because 3 base pairs are deleted. So what that means, so if you can see, if you were to rewrite the sequence of the mutant gene coding strand without the X's, you would find that 510 and 511, those codons would remain the same. And so since that's the C-terminus sequence, that would mean there's no change in the C-terminus sequence. Does that make sense?
Yes, it does.
Okay. And then for the second part, I have to go back to that genetic code page, that genetic code chart, which is on page 220 for the Kaplan books. Or I have the hard copy version. I'm not sure if that lines up, but it should be figure 7.5. And so you kind of have to figure out which residue will be deleted. So looking at 507, you have ATX.
And what page were you looking at?
220, if you have the hard copy.
Okay.
And so one thing you have to know for this question is that the coding strand is the same as the coding strand is more or less the same as the mRNA strand that's made. So coding strand is DNA, and then the mRNA is is like basically the same thing but with U's.
Mm.
So in that case, for 507, we have ATX. So that would be AU blank. And according to this chart, so oh, sorry, it's actually 508. So it would be
UU.
UUU or UUC. So like pretty much it's more likely that phenylalanine was deleted than leucine, if that makes sense.
Okay, I got it.
Okay.
Thank
Awesome.
you.
You're welcome. Any other questions? We'll maybe take one more before we move on.
I had a concept question, if that was okay.
Sure.
I was gonna ask you for like coding and non-coding strands, right?
Mm-hmm.
What is the like appropriate direction that like both of them are read in? Like which like the coding is like sometimes I get confused like what direction the coding strand is read in and what the non-coding strand is read in.
I have that on one of my slides for the talk.
Okay.
Yeah. So would you be willing to wait until I get there?
Yeah, that's fine.
Okay, awesome. Okay, should we move on to the next section, Pranava?
Yeah, that sounds good. I'll go ahead and share the PowerPoint again.
Yeah, you can just take over.
Yeah. Okay, let me know if you guys can't see my screen. So this is the first warm-up question. We just have 4 discrete warm-up questions. So the question is, the DNA of telomeres can be most accurately described as non-coding and highly repetitive, single-stranded, self-complementing, able to encode multiple unique proteins, or easily cleaved by restriction enzymes. I'm not sure how we want to do this. If anyone is willing to unmute themselves and kind of volunteer their thoughts, or maybe put the answers in the chat. What are you guys thinking of?
I'm unable to use the chat, but I think it's A.
I agree as well.
Sounds good. Does anyone want to explain? Sorry, I cannot see your chat while I'm presenting. So Karen, can you let me know if there's anything in there?
A lot of you guys got it right. All A's.
Ironically, I just learned this in genetics, so that's another reason why.
I think it's A from my understanding because telomeres, like they prevent the degradation of like the chromosome. So, and then since they're non-coding, because each time like DNA is replicated, the telomeres are they're shortening.
Yeah, exactly. So it's kind of like a protective element. So as you just explained right now, after each round of replication, the telomeres get shorter and they're non-coding so that even if they're lost, there's no loss of genetic material. And then it's always good to like eliminate the wrong answer as well. So for B, it's DNA, so it's not single-stranded. And then C, it doesn't encode proteins because they are non-coding. And then D is not relevant to the question, is how I would think about it the easiest way. Okay, and then the next one, which of the following nucleic acid complexes would undergo correction by the DNA mismatch repair system? So I think you guys can put it in the chat. I'm going to go ahead and show the answer now, which is C. And then, so this is a fairly straightforward question. You're basically just looking for the mismatch. So the challenge of this question is not necessarily the concept, but like literally digging through these like 20 base pair long sequence to find this error. So sometimes you will run into stuff like this. So you got to prepare for like that frustration. Yeah.
I have a question.
Yeah.
On answer choice D, C is paired with A. Does that have to do with like wobble position?
Yeah. Okay. That's a good point. So, yes, so I guess we'd find our start code on here. So then that way we will look at like each first codon, or sorry, first base of each codon. So this is the first. So then this would be the first. And then so this. Sorry, I didn't notice that earlier. Can I get back to you on that one? That's a really good point, though.
Yeah, of course.
Yeah, I'll put it as in the like Teams chat, and also I'll do that. Sorry about that.
No, you're good. Thank you.
Third question.
Okay, so this is a genetics question, everybody. Surprise. So assume L410 is an acquired mutation that occurs only in hepatocytes of adults who were exposed to a certain mutant gene. A woman with the L410 mutation has a child with an unaffected man. What is the probability that this child has the L410 mutation? So go ahead and put your answers in the chat if you think you know. Okay, I'm seeing some B's. I'll give you a few more seconds and then you can reveal the answer.
Oh, sorry about that.
It's okay. Oh, okay. So the answer is A. I know I saw a similar question to this when I was studying and it tripped me up. So the trick to this question is the word is two words. acquired mutation in hepatocytes. So that makes it a somatic mutation. So when you acquire a somatic mutation, which is a mutation anywhere other than your germ cells, so like your sperm and your eggs, so cells that can't be passed down to your offspring, that means that there's no chance of passing it on. So that's what a somatic mutation is, as opposed to a germline mutation, which is acquired in your sperm cells or eggs, which can be passed on to your offspring. So that's the answer.
I have a question. Could you also say, like, your reasoning is great, but like, could you also use that it also doesn't give like a 0.5 as a probability, and there's no talk about like it's not like recessive or dominant or anything. So like that's also a good hint that it's 0 instead of the other answers.
Yes, that's an amazing point. Thank you. And then any other questions about this? Okay, never mind. We'll move on to the next one. If you have any questions, just let me know later. So this is a tiny one. Sorry if you guys can't see this. So researchers are screening bacterial chromosomes for mutations affecting protein function. After using DNA sequencing analysis and analyzing the gel electrophoresis results, they identify a sequence with a single point mutation in which the fourth nucleotide has been changed from adenine to cytosine. And so you can see in the little sequence that they give us that the fourth nucleotide is indeed cytosine. So which of the following gel electrophoresis diagrams obtained from DNA sequencing analysis represents this sequence?
Is it B?
Yes, the answer is B. Is everybody ready for an explanation? Okay, so this question is fairly simple, a lot simpler than the last one, not as tricky. So the thing you have to know about DNA sequencing is that when they give you the way that the sequence is written, it's basically written in the same way in the gel electrophoresis. So you should expect to find the same nucleotides written from 5' to 3'. So if you can see here, it's TTACG, and that's the only answer choice that matches the sequence correctly. Carmen, you have a question?
Yeah, is this under lab techniques section to like understand gel electrophoresis, or is this like in bio section? Like, I just want to know like which section is actually in this part. It's because we had to learn lab techniques.
Yeah, lab techniques are included in this section. It was mainly in chapter 6, and they don't go over it too much. So I definitely suggest watching more videos about it.
All right, thank you so much.
Could you quickly describe the difference between why B is correct and D? The difference between
B and D?
Yeah, the answer choices.
Oh, okay, so D is kind of the inverse in B, in that the DNA sequence has the nucleotides written from 3' to 5', while the correct answer B has it sequenced correctly from 5' to 3'. Does that make sense?
A little.
Okay, so
Can you explain for me again why B is the choice?
okay, so we have the sequence here. We have the sequence 5' TTACG 3'. And so what you would expect from DNA sequencing is So you have the sequence written horizontally in the question, and then you have kind of the sequence written vertically in the answer. And you should be paying attention to the right side of each square to see that 5 primes at the bottom and 3 primes at the top. So you should keep that in mind. So all you have to do is kind of match up, is kind of follow the sequence from 5 prime to 3 prime with those same nucleotides. So starting on the bottom, so 5 primes on the bottom. So only choice A and choice B have T as the first nucleotide. Does that make sense? Is everybody following so far?
Yes.
Okay, and then
Yeah.
the next one is T, and so B is the only choice that has 2 T's in a row. So that makes it the right answer.
Okay, so you just basically had to read it from the bottom up in the same direction as it was going horizontal.
Yeah. There are going to be harder questions you might run into later on in different materials that might kind of ask you to convert it to that they might give you like an mRNA sequence, or they might give you like an anti-coding strand. So it'll be a little bit trickier. So this is kind of the simplest version of this question.
Thank you.
Hi, I have a question. Can you tell me where I could find this kind of questions to practice?
So Pranava found these questions on UWorld.
UWorld, okay, thank you.
Yeah, but it does cost a bit of extra money. It costs around under $300.
And that's for 3 months, right?
You can choose how long you can subscribe for.
Okay, thank you.
Mm-hmm.
Here's also like a sorry. I'm gonna also put like Khan Academy DNA sequencing like overview, and it has some questions at the end of that. So this is a free resource. Sorry, I'm lagging a little bit.
And then I see whoever MS6AS is. Did you still have a question?
No, it was actually already answered. Thank you.
Okay. You can lower your hand if you like.
Oh.
I can't put that link in right now, but I'll do it at the end. Sorry about that. Are we all ready to move on to the brief like topic review? Okay. So I'm gonna try to like breeze through this as much as we can, and then if anyone has any specific questions, can interrupt us. So looking at the monomer of a nucleic acid, it will be called a nucleotide, which is the combination of a base, a sugar, and a phosphate. Sometimes you'll also see nucleoside, which is basically a nucleotide minus the phosphate. So this top right image is an image of the 5 nitrogenous bases. So thymine, cytosine, adenine, and guanine are found in DNA, and then uracil is RNA only, and it replaces thymine. And here are the 2 sugars. The difference between ribose in RNA and deoxyribose in DNA is that deoxyribose is missing an O in the second carbon. Another thing to know is the nucleic acid polymerization. So we have these individual nucleotide monomers. How do they polymerize to form a long chain of DNA? Is through this phosphodiester bond. So the phosphate group on the 5' carbon of one nucleotide is going to bond with a OH group on the 3' end of a different carbon. Of a 3' carbon of a different nucleotide. This is going to be a dehydration reactions, which means it's going to lose a water molecule, as all polymerization reactions are, and it's going to form this phosphodiester bond. And nucleotides have direction. So the 5' end is what is called of the end that has the phosphate group sticking up. And then the 3' end is what is called the one where the OH group is sticking down. So this is the 3' end because here's your OH group sticking out, and then the same thing on the other side, it's the OH group sticking out, and here's your phosphate here on the 5' end, and here's your phosphate here. So DNA is also anti-parallel, which means one strand runs 5' to 3', and the other strand runs 3' to 5'. And here's just another picture of what the carbons look like. So this is what we call carbon 1. This is 2. I've connected to the OH on ribose, and then just an H on deoxyribose. And here's your third one, which is this OH. Like, don't be confused by that. And then this is your fourth, and the 5th carbon is attached to the phosphate. In a brief overview of the central dogma in biology, DNA is transcribed to make mRNA, which is translated to make protein. And a little bit like a side information off that is that DNA is also replicated to make more DNA. And the difference between the central dogma.
A quick question.
Yeah.
Can RNA be replicated?
RNA, I do not think it can be replicated. So what happens is DNA expression is turned on and off. So when DNA expression is turned on, more mRNA is made. When DNA expression is turned off, mRNA is not made, but you can't use mRNA to make more mRNA. It has to come from DNA.
Okay, thank you.
So the difference between the central dogma in prokaryotes versus eukaryotes, so the basic transmission of information is still the same. We're still go from DNA to RNA to protein. The locations of these processes are a little bit different. So in bacteria and prokaryotes, transcription will occur in the cytoplasm because prokaryotes, remember, don't have any membrane organelles. But in eukaryotes, transcription occurs in the nucleus, and then the mRNA is sent out of the nucleus to be translated. So translation always occurs in the cytoplasm, but whether it be in eukaryotes or prokaryotes. And the other difference is that in eukaryotes, there's something called mRNA processing, which we have slides on later, I believe, whereas in prokaryotes, there is no mRNA processing. So you get an mRNA, and that's what you translate, but in prokaryotes, but in eukaryotes, you get your mRNA, which is actually going to be called your pre-mRNA, and you do a bunch of stuff to it, remove introns, add a poly-A tail, and a 5' cap, and then you have your mature mRNA ready to be translated. So there's also a couple of repair mechanisms when DNA replication occurs erroneously. So that means like if you have a base mismatch, such as like having an A bound to a C as we had in that earlier problem. So there's kind of like two major ones to highlight. So the DNA polymerase itself, which carries out DNA replication, has exonuclease activity, which means it can cut out bases that are incorrect. So that's one of the main functions. That's one of the main ways repairs happen. So this is called like proofreading ability of DNA polymerase, if you ever see that term come up. So as it's polymerizing, as it's replicating the DNA, it adds a wrong base, but then it'll recognize it and go back and fix it in with the correct base. The other repair mechanism is called the DNA mismatch repair. So they're basically like protein complexes that you don't need to know too much about. But there's a couple types, like single base mismatch or repeat mismatch. And I think the primary thing for you to know here is just know that there's like a complex of proteins that'll cut out the DNA, and then like DNA polymerase and DNA ligase will fix up the cut to fix the DNA again. Okay, and then this is, okay, Karen, are you ready? Do you want me to give you the screen?
Sure. Give me 1 second. Okay, can everybody see my slides?
Yes.
Okay, so now we're going to go over kind of some more details about the central dogma, starting with replication. So I have this diagram of DNA replication here. So basically, there's a lot of enzymes involved. And the first thing that happens is that the DNA needs to be unwinded using an enzyme called helicase. And then topoisomerase is an enzyme that functions to kind of, when it's unwinding, there's kind of a lot of strain that happens, and topoisomerase kind of helps to relieve that strain as the DNA is unwinding. And then afterwards, you have the RNA primer that's placed into the open strand of DNA. And then you have RNA polymerase that comes in and replicates from a 5' to 3' direction. And DNA replication is also bidirectional in that there's a leading strand that's kind of going this way, that's replicating this way, and there's also another lagging strand that's replicating kind of this way, like so in the same direction, but like, oh, can you still see my screen?
Yeah.
Okay. And then for the lagging strand, an important thing you need to know is that DNA bases are added using things called Okazaki fragments. So it's kind of a section of bases kind of put in at one time in kind of the lagging strand's direction. And also another thing I wanted you guys to know is the fact that DNA replication is semi-conservative. So this is kind of a diagram of the experiment done by Messelson and Stahl. So they took two different kinds of, they took one whole DNA strand that was made with 15 nitrogen 15 bases. They're all marked with nitrogen 15, and then they used nitrogen 14 to add to label their new bases, so when they're being made, and they wanted to find out how DNA is being replicated. So there's semi-conservative, I believe there's conservative, and there's also, I forgot the other name. I want to say liberal, but that's probably just like, there's 3 different kinds, and the right one is semi-conservative. That's kind of the point. And the reason why it's semi-conservative is because one strand is old after replication, and the other strand is new. And so as you keep going, so for this strand right here, this one's old and this one's new as well, because this was the old strand. And this one is the old strand right here, so this is the old strand here, and there's a new strand here. So if that makes sense. So that's the semi-conservative replication. And DNA replication differs between prokaryotes and eukaryotes. There's a lot of differences you got to know. One of them is that eukaryotic replication occurs in kind of a linear DNA fashion because that's the structure of most eukaryotic DNA. As opposed to prokaryotic DNA, you're dealing with circular DNA. Because of the structure, in eukaryotes, you're going to see multiple replication bubbles form until you have, and the replication bubbles are kind of eventually going to come into each other. And so you'll have, in this case, you'll have 4 different strands at the end. And then for prokaryotes, you have one origin of replication, and you have the replication forks right here. And then the replication forks are kind of the corners of each bubble right here. And also another thing I wanted to note is that for replication, the enzymes tend to be slightly different between prokaryotes and eukaryotes. As I said before, DNA is synthesized from 5' to 3', and then lagging strand is discontinuous, but that's kind of across the board. Or that's mainly, the lagging strand is mainly, I forgot what I was going to say, I'm sorry. But if you can notice here, you have the pink enzymes for prokaryotes and the yellow enzymes for eukaryotes. It's kind of a small detail, but that's kind of just how intricate these differences are. So that's kind of my point.
Should we memorize the differences between enzymes?
It's not that important. Yeah, it's not too important, so I wouldn't worry about it too much. Just know that the enzymes are different.
Okay, thank you.
You're welcome. And then, so this, I apologize, this is the only slide I have on the biotechnology. I decided to go over 2 kinds of recombinant DNA techniques, which is kind of gene cloning and also PCR. So we have, so this is an example of DNA cloning. So you have kind of the vector DNA, which usually comes from a bacteria, and the donor DNA, and you use this thing called restriction enzymes. So the word restriction, it's like you're restricting the length of the strand. So it's a way of cutting it. So the restriction enzyme basically cuts the ends of both the donor DNA and the vector DNA. And if you notice, it's kind of like there's like, it's not exactly like it's kind of like a Z. So you have like the Z right here and kind of the Z right here, if you can keep track of that. And these little nucleotides hanging out are called overhangs. And so once you cut those, you'll be able to kind of, you'll be able to kind of put them back together and using an enzyme called DNA ligase. And so ideally, the overhangs of each one should match up with each other. And so that's why you have these overhangs matching up with each other. And so the overhangs are sealed together. And from there, you can introduce the DNA into bacterial cells, and those cells replicate and divide. And so in that case, the DNA itself is replicated too. Another thing I wanted to go over is PCR, that stands for polymerase chain reaction. So you have your double-stranded DNA, and this happens a lot in the lab. So you have your denaturing stage, which kind of separates the strands, and it's at a very high temperature, almost 100 degrees Celsius. And then you have your annealing stage, in which a primer is introduced so that the matching strand can be synthesized at the annealing stage. Annealing means kind of the joining of 2 strands, and so that's what that means. And that's around 50 to 56 degrees Celsius. And then the extending stage is kind of where the replication occurs, which is 72 degrees Celsius. And by that time, after that stage, the DNA should be replicated, and you just repeat it all over again. And so that's a very quick way of getting more DNA. And some other things I don't have time to talk about right now, but was mentioned in the textbook. There's DNA libraries or cDNA libraries. It's kind of like a library of kind of like a genome library. And then hybridization and annealing, I kind of went over annealing. Hybridization is kind of the same, has the same meaning, but it's used in different cases. And then blotting is used to detect sequences in DNA. So a lot of times fluorescence is used, and then there's a difference between southern blotting and northern blotting and western blotting based on what kind of substance you want to illuminate, whether it's DNA or RNA or protein. I might get it wrong with northern versus western. It's been a while. And then DNA sequencing is similar to the question that we had in the warm-up. And then DNA versus RNA. This is so there's two main things you should know. DNA stands for deoxyribose nucleic acid, while RNA stands for ribonucleic acid. So deoxy means that it's missing oxygen, and where is DNA missing its oxygen? It's missing its oxygen on the 2' carbon. So if you guys aren't familiar with organic chemistry, this is the 1' carbon, 2, 3, 4. 1, 2, 3, 4, 5. Yes. Because this is a ribose. Sorry, I thought this was a carbon. My bad. So that's 1, 2, 3, 4, 5 carbons. As opposed to ribose, it still has its oxygen on the 2' carbon. Another difference is that DNA has thymine, and while RNA has uracil, they both go with adenine. If you guys don't know, this is adenine, and this is guanine, and this is cytosine. And generally, DNA tends to be double-stranded, and RNA tends to be single-stranded. When we get to our next lecture, which is viruses, the RNA rules will kind of be thrown out the window, so just keep that in mind. But in terms of the central dogma, just think of RNA as a single strand, okay? And before I go into transcription and translation, I kind of want to just really quickly go over different types of RNA, because I saw a lot of different kinds of RNA while I was studying. So mRNA is the only coding RNA, and everything else is non-coding RNA, because mRNA is eventually used to code for the protein. So that's the only one that's used to code for the protein. Other kinds of non-coding RNA, you have the transfer RNA and rRNA, which are both used in translation. We'll talk more about that a little bit later. And then we have all of these small non-coding RNA. That's what it stands for. Generally, in my studies, these tend to be used post-transcriptionally. So they tend to so for example, microRNA, it has the ability to block translation altogether. While snRNA, it's used for splicing, which is part of post-transcriptional modification. And also in the textbook, they had this RNA called hnRNA. It's basically pre-RNA, pre-mRNA, and they just have this name. So just in case you guys are confused, it basically means pre-mRNA, which is eventually made into mRNA. So transcription. So going off to the question that one of you guys had, so we have the coding strand, the template strand, and the RNA strand. So what we were saying during one of the questions that we had is that basically the coding strand and the RNA strand are identical, except for the thymines and the uracils. So that's kind of something that you can keep in mind that's pretty important to remember. And the template strand is the strand on which RNA is transcribed. So the coding strand, there's the coding strand which has almost the same mRNA, the same RNA code, if that's a good way for you to remember. And then the template strand is the template on which the RNA transcribes. And the RNA is read, so the template strand is read from 3' to 5', and the RNA is synthesized from 5' to 3', if you can see how this dynamic is going. So the most important thing is that the RNA is synthesized 5' to 3'. And then I mainly included this this picture to show that for RNA, it does require a little bit of like unwinding that happens at the same time. So the enzymes helicase and topoisomerase you can also consider in transcription as well. And then there's three main sections to transcription. There's initiation, where the RNA polymerase, so that's used in transcription, like we said in the question, is binded to a promoter, which is usually upstream, so upwards. So this is upstream and this is downstream for the most part, if you guys, for terminology. So the promoter is located upstream of the DNA that needs to be transcribed, and transcription begins at the start site. And then elongation is when the RNA is being synthesized, and termination is when it reaches its termination site, in which case the strand is released and the process is finished. And some post-transcriptional modifications, there's three major ones.
I have a really quick question.
Yeah.
So if you go back to the last slide, you said that the coding strand plus the template strand were identical, but from my understanding, the only difference is that the coding strand has those uracil nucleotides, right?
So the coding strand and the RNA strand, the mRNA strand, are almost identical, except for the coding strand is DNA, so it has thymine, and the mRNA strand has uracil.
Okay, so you said the coding strand is DNA?
Mm-hmm.
I thought the template strand was DNA, though.
They're both DNA.
Okay, really? Because I thought the template strand was referred to as the non-coding strand.
There's actually a lot of different names for it. So template strand, and another name for it is non-coding strand. Another name for it is, I believe, antisense.
Right. So then just with that, then how could the template strand and the coding strand be the same thing? Then
They're not the same. They're complementary to each other.
Okay.
Sorry, so just to clarify, like it's the template strand because it's a template that RNA uses.
Yes.
Okay, thank you.
Okay.
All right.
Okay. So moving on to the next slide, if you have any more questions, feel free to save it till the end. So there's three main post-transcriptional modifications that you need to know. So the first thing is splicing. So splicing is the process of taking basically, so you have the pre-mRNA transcript here, and it has exons and introns. So the exons, so this is how I remember it, the exons are the parts of the mRNA that are expressed. So those are included in the final mRNA strand. The introns are basically thrown out. And so the process of getting rid of the introns and putting all these exons together is called splicing. So that's the first post-transcriptional modification you need to know. Second one is that there's a 5' cap of guanine, and they're 5' cap of methylguanosine that's added to the 5' section of the mRNA. And the third one is at the 3' end of the mRNA, there's a poly-A tail, so adenine, polyadenine tail that's added here as well. So number 1 is splicing, number 2 is the 5' cap, number 3 is the poly-A tail. And another thing I wanted to talk about is that there's more than one way that you can splice things together. So that's called alternative splicing. So for example, this might be like the primary way to splice it, but they also use, they can also put it in this combination to make different proteins. And so this is how you can use more than one pre-mRNA to make multiple proteins. So that's alternative splicing. And then this is, so right now I'm going to talk about the regulation of transcription in prokaryotes. And the main way that's done is through operons. So an operon, so the model was proposed by Jacob and Monod to explain gene regulation in prokaryotes. And typically an operon should have all 4 of these components. It should have a regulator gene, which tends to make the repressor if a repressor is involved, or a repressor is usually involved. And then there's the promoter section. There's a promoter section. There's an operator section where the repressor binds to and affects whether or not something is transcribed. And then 5 is the genes that need to be transcribed. So I guess there's 5 things. And then there's 2 types of operon. There's 2 types of ways that operons function. And so this one is called an inducer-repressor complex. So this is kind of an inducive way. I forgot the name of it. There's like the inducing operon complexes, and then there's also the kind of repressor kinds. So the way that the inducers work, so an inducer is kind of an exogenous substance that comes in and binds to the repressor. And so in normal cases, the repressor would bind to the operator and it would prevent transcription. So the repressor represses transcription. The inducer, by binding to the repressor, prevents the repressor from binding to the operator. And so in that case, it induces transcription. So the inducer induces transcription. One of the most popular inducer operon systems is the LAC operon, so which stands for lactose, and lactose and glucose are involved in that one. That's a pretty popular one. You'll probably run into it while you study. And the second kind of operon structure is the repressor-co-repressor model. And so in this case, the repressor has a co-repressor that kind of mesh together. So if you can notice the shape of this, it can't bind to the operator on its own. And so without the co-repressor, the transcription occurs. But when you combine these two to kind of make the shape that fits this operator, when the co-repressor and the co-repressor bind together, they both combine to repress transcription. So that's kind of the repressor-co-repressor complex prevents transcription, while the inducer-repressor complex induces transcription. And for repressor-repressor complex, that happens a lot with, in the textbook, there is a tryptophan example. So that's that. And then regulation of transcription in eukaryotes is a lot different. So the main things I want to focus on here are the enhancers and the transcription factors. Usually the enhancers and the transcription factors work together to enhance transcription, which basically is like increasing it. One thing with eukaryotes is that the enhancer can be located pretty far from the promoter. If you notice in the prokaryotes, the promoter is quite close to the regulator is close to the promoter. So the enhancer here is pretty distal from the promoter. So distal means far. And generally, enhancers enhance transcription, and transcription factors, they bind to enhancers to kind of really enhance transcription. So without the transcription factors, the enhancing wouldn't really occur. In addition to enhancers, there's also things called silencers, and it's pretty self-explanatory. They do the opposite. So when transcription factors bind to silencers in this complex, transcription is silenced. And also, so this is kind of at the DNA strand level, and this is kind of at when DNA is all wound up, and they're wound up into things called histones. So histones are, this is the DNA. So this purple stuff is the DNA. And histones are proteins that DNAs wrap around to kind of pack more tightly together. And this kind of format is called chromatin. And there's 2 ways you can modify chromatin to affect transcription. There's histone acetylation, which in effect kind of, when you add an acetyl group to the histone, it kind of loosens up the histone, and it allows the DNA to kind of be regulated, to kind of be manipulated, and in that way, like expression increases. And then there's another one called DNA methylation, is when you add methyl groups to the DNA, and it kind of makes it pack even more tightly together. And so it's harder to, it decreases transcription in that way. It makes it harder to transcribe. So there's histone acetylation, which increases transcription, and DNA methylation, which decreases transcription. And then with translation, so there's 3 steps to translation, similar to transcription: initiation, elongation, and termination. So with initiation, you have your tRNA, your transfer RNA. So it already has a methionine, and it binds to the starting site of this mRNA here, which has the nucleotides AUG, which makes sense because AUG codes for methionine. And the mRNA is kind of placed on the small ribosomal subunit, and then after that, the large ribosomal subunit comes in, and then it places the tRNA at the P site. And so this whole thing with the small subunit, the large subunit, the mRNA, and the tRNA, this all makes the translation initiation complex. And an important thing to note is that this requires GTP, which is a form of energy. The next step is elongation. So this is the entire cycle for elongation. So the first thing is that the codon is recognized so you have a new tRNA come in the A site, and the codon is recognized here. So that's step 1, and that requires GTP as well. Step 2 is peptide bond formation. So basically this chain right here is going to hop on over to the tRNA that just came in. So you have this little thing hopping onto this, and so in that way a peptide bond is formed, and a peptide bond is an amide bond. So if you remember organic chemistry, so that's step 2. Step 3 is translocation, which also requires GDP. So you're kind of kicking out this old tRNA to the E site, which stands for, I think of it as the exit site, and the A site, and then so this one is going to go to the P site and be kind of the new resident here. So that's translocation. And then after this step, you're ready to elongate all over again, because a new one's going to come in, and then the peptide's going to be shifted here, and then the old tRNA is going to get kicked out. So that's elongation. And then termination occurs when something called a release factor enters the A site, and it matches to one of the stop codons, UAG, UAA, or UGA. And so this is a release factor binding to one of these stop codons. In return, it kicks out the old tRNA, and with some GTP added, it disassembles the entire complex. So that's when the translation process is officially ended, when this whole thing is broken apart and the polypeptide is released. So translation in prokaryotes versus eukaryotes. So first of all, they have different ribosomes that are used in translation. So if you can imagine like the mRNA being here and kind of the tRNA being in here, but they have different sizes. So the S stands for a unit called Svedberg, which is like, I think it's like some sort of like sediment units, something like that. And the way I remember this is that the eukaryotic ribosome has all even numbers for both their subunits and their total weight of the ribosome. So even numbers 60S for the large one, 40S for the small one, and 80S for the total. And as opposed to prokaryotic ribosome, it's all odd numbers. So that's 50S for the large ribosome, 30S for the small ribosome, and 70S total. Another difference that you should know is that eukaryotic mRNA is a term called monocistrionic, which means that translation starts at only one place in the mRNA, while prokaryotic mRNA has multiple translation start sites, so it's considered polycistrionic mRNA. So monocistrionic, one start site, polycistrionic, multiple start sites. And then also another important thing I should note is in prokaryotes, transcription and translation occur simultaneously. And so the mRNA is kind of used immediately. There's no processing step in between transcription and translation. While in eukaryotes, you have them occurring in different places and not at the same time, because the transcript needs to be modified before being translated. And here's a very nice diagram of a lot of post-translational modifications. So once you got your protein right here, there's a lot of things you can do to it. In the book, they mentioned phosphorylation, where you add like a phosphate to pay attention to one of these amino acids, serine, threonine, or tyrosine, which is important because they all have OH groups. So you can only add a phosphate group to anything with an OH group. There's also glycosylation, where you can add a sugar. Ubiquitination is kind of like you're targeting it for destruction, basically, for degradation. Lipidation, you add a lipid group to it. Methylation, you add a methyl group. A lot of them are self-explanatory. And that's it for review.
We're not going to have time to get to the passage tonight, and we don't want to hold you like way longer, but I believe you'll get the slides after we log off tonight. I'm not sure when, but you will get it, sometimes within the next couple of days, and the passage is attached in there. And what we're going to do is we'll type up the answer explanations like in the notes section of the slides. So that way you guys can see all of those. And if you have any questions, you can always like reach us on Slack or now Teams or email us. Does that sound okay? Does anyone have any questions?